And I think you've heard from Sean that there are many variables in these experiments. I hope you appreciated that. And site of experiments have many moving parts. And this slide deck really came together because we'd get phone calls from people and they'd say, can you run my samples? And 2 weeks later it'd be, have you run my samples? And the answer was a big no. And so what I'm going to do is sort of give you an overview of all the variables, not all, I'm sure there are things I haven't thought of, but the variables that you need to consider so that you have data, generate data that is technically rigorous. And so, whoops. Oh, slide's not advancing. Don't know why. Switch. Right arrow. Still not. It's advancing on my screen, sorry, but not on the projector. Oh, right. Sorry. Yep. Okay. Yep. Okay. So you've sort of, this is sort of another version of what you saw from Sean, which is the workflow. So the workflow for mass cytometry is very comparable to fluorescence-based cytometry. So that's really good. And those are just experimental variables that are just the same. And so that is the fact that we interrogate cells to look at intracellular responses. So that means the antibodies, the fixation and the perm protocols are the same. The staining with antibodies here with fluorochrome conjugated antibodies, you pass through the laser, you look at and you find cells that you wouldn't have found if you hadn't added these stims. And what we're doing here is now we're replacing the fluorochromes with metal conjugated polymers and passing them through the argon inductively coupled plasma and getting pretty comparable data. And you already saw that from Sean. So I just wanted to actually just give you an overview of how the time is divided and what to expect. So the major bulk of the time is at the front end and the back end. So the front end is the experimental design and reagent validation, and I'm going to go through that. And that really is months, and it can be a year. And I'm telling you the year from my own personal experience. And actually, the experimentation can be just 1 to 3 weeks. And then, given the fact that one is looking at single cells, hundreds of thousands to millions in up to 40-dimensional space, that analysis clearly takes time, and that can be months. So what are the considerations for the experimental design? And the first thing is, people do come and they say, can you run my samples? And I'm like, what would you like to look at? And while on the one hand, mass cytometry is really great for surveying the biological landscape, you do have to have some idea of what it is you want to look at and what questions you want to answer. And so what's the background, the biology or otherwise, to drive your questions? And then, what's the perfect experiment that you could design? If you had your way, what would it be that would really give you the answer? And most of the time, a lot of the time, that is not possible, especially when you're looking at, in vivo and clinical situations, which of course is my own personal excitement, is what we can do here, leverage the new layers of information to really make an impact for the benefit of patients. And so when I say the perfect experiment, clearly with solid tumors and my own work in ovarian cancer, I don't know how that tumor is behaving in that patient, but I have to adapt the conditions, which means making single cell suspensions, checking the validity of everything, and that I consider to be a compromised experiment. Nothing you can do about it. So think about, these are some of the other things to think about. What is the appropriate model system? So what's the application? And here are some examples. And again, this is not exhaustive. Are you interested in pathogens, viruses, bacteria, cancer, immune disease, immune monitoring, therapeutics? What will be the models that you're going to use? Are you going to do a murine system? Are you going to do a non-human primate, a human, or ex vivo, which would be cell lines? And here are some of the samples that you could use. You can use whole blood, you can use peripheral blood, monocytes, say, bone marrow, solid tissue from wherever, but to be suspended, generate single-cell suspensions, tumors, and cell lines. And then comes the exciting part, is planning the experiment of all the things that you can look at. And so what's really cool is, and I think that is what excites me, and probably I speak for a lot of people, is these simultaneous measurements that can span a broad range of biological function. So we can look at the phenotype, which is surface markers to tell you, delineate the specific cell subset, and then simultaneously you can measure the cell cycle. You'll be hearing about that from Greg Behani tomorrow. DNA damage, apoptosis, metabolism, survival, proliferation, immune responses, transcription factors, pluripotency, cytokines, on and on and on. And one thing that I've always was very intrigued about was the fact that with the cell cycle, you can delineate all phases and you can actually find out what is happening in those phases. And I'm sure Greg is going to talk about that more tomorrow. Well, he will. And then, so what we have here is your antibodies can be divided into two sets. Ones that recognize extracellular proteins, as I mentioned, to delineate specific cell subsets, and intracellular proteins that can tell you about cellular function. Okay, so what are the key reagents? So your experiment and your data, I hope I'm not telling you anything that is a surprise, is as good as the reagents that you put in. And therefore, that has got to be highly regulated and controlled and verified for quality. And so those samples are cell lines and primary samples. The choice of the perturbants or stimulants, and then the antibodies have all got to be validated. And for those, the antibodies are against the surface markers, the functional markers. Things to consider are the antibody clone selection, your titrations, the metal assignments, and the conjugations. And then, of course, there are other variables within the experiments, such as the titrations of the stims and the kinetics, fixation conditions, permeabilization, the washes, and where the hell can you take a break? And so, as I mentioned, the workflow of fluorescence and mass cytometry are very, very comparable. So what's really nice, if you don't have a cytoff, but you have a collaborator, what's really nice and helpful to everybody, especially to you, is to perform as much background experimentation as you possibly can, because chances are that the things that you see by fluorescence, well, are that it will be directly transferable to mass cytometry. So, as I mentioned before, what's the biology? It really is good to know that, and be surprised how many people have asked to do an experiment, but they actually haven't thought of the question they would like to ask. And so we have a Flow 101, which I think Sean was nixing out, but I'm going to actually put it back in another slide.
I didn't want to nix it. I just, I was looking at the lecture schedule, and there was a lot of.
I'm teasing
you. A lot of cytops.
Don't get defensive. And so what you can do, so the Flow 101 will just sort of tell you something to see if you haven't had much fluorescence flow cytometry experience. It'll just get you up to speed with being able to stimulate cells and looking at extracellular, intracellular antibodies, and doing a little bit of fluorescent of compensation to get your answers. And then you'll probably, in your specific project, you'll have new antibodies. And so it's really nice if you can validate and find the clones, validate them on fluorescence. And then the stim, the fix, and the perm conditions, again, they're not going to change from going from fluorescence into mass cytometry. And then you are ready for thinking about doing a mass cytometry experiment. And so there will be some differeses, of course. And those differences are really based on the fact that the antibodies are now conjugated to metal chelated polymers. And so the key points are, when you build and design your panel, are the metal assignment. And one thing is that the metals are actually enriched. They're not pure. And so even though you could, in effect, you could probably put any antibody on any metal, you probably get some good answers. But there is a metal purity matrix. And so considering that, and so there's sort of a little bit of spillover. It's nothing in comparison to fluorescence. But nevertheless, if you've got, for example, a very abundant protein and a less highly expressed protein, you want to be careful how you assign the metals, because you don't want the abundant protein the signal from the abundant protein spilling into the signal from the less abundant protein. And that's kind of common sense, and so you can do these tweaks and you can really optimize your panel. And then there are a lot of metal conjugated antibodies commercially available from DVS, and they're actually based on Sean and Aaron's science paper, and so you can just buy them off the shelf. However, they don't have every antibody on every metal, and so if you buy them or choose to buy them from them, at the moment it limits the flexibility of how you can design the rest of your panel. So that means you have the opportunity with some MaxPar kits to do your own conjugations. And again, this is based from the protocols developed here with Sean in particular. And so you can do your own metal conjugation. And then, but regardless of whether you purchase it or you do it yourself, you should titrate it. And yes, I know the vendor will say, yes, I recommend so-and-so this amount, but I'm of the school of thought that one should always just see for yourself. I'd never take anybody's word. Don't believe them. Then, of course, there's ready for the sample prep. Then a number of places. This is where you could take a break. So you can actually do the whole thing and be totally exhausted, or you can actually do the stim, and you can fix and you can wash out the paraformaldehyde, and you can freeze at -80, and then you can run the samples after that, or ship to somebody who does have a site off. So here I'm just going to go through some of the key backgrounds, and these next slides, I apologize, are sort of listy, but I figured that, rather than going through them in intimate detail, you'll have them as a reference, and so that's really the point of this. So as I mentioned, you can confirm the biology of interest using fluorescence. So it's really nice if you don't have a mass cytometer and you can say, look, this is what I want to do, and here are the antibodies I'm interested in, the proteins I'm interested in probing, and I've already figured out how they work by fluorescence. And so you want to, and again, with the fluorescence foundational work, choose the relevant cell lines. They might be primary cells, the relevant stims, the antibodies, titrations and optimization, testing different chromes, really important. I'll show you an example of that, which I put in later. It's not in your handout. And then cell staining for surface markers on fresh cells, on fixed cells, and intracellular signaling proteins. And yes, I just want to tell you, the buffer conditions and all these conditions, they do affect the recognition of your epitopes by the antibodies, because a lot of these things are denaturing. Freezing is denaturing, paraformaldehyde is denaturing, but you can't have a perfect condition for every antibody. And so at the end of the day, the panel and how you choose to work on it is the result of compromises. And so what sort of looks bad, you might have to take a few hits, not usually, but not usually hits that are too terrible, but it is about compromises. And so again, if you are looking at your in your background work, antibodies to delineate specific cell subsets, and for example, a simple example in peripheral blood mononuclear cells, CD3 for T cells, 19 for B cells, 33 for monocytes, and then antibodies for the intracellular proteins. These stats are activated by a variety of stims, as is ERK and phospho-AKT. And you can do some sort of small titrations with the fluorescence. It's just nice to get an idea of how good the antibody is before really going into detail when you have got your metal conjugated antibodies for mass cytometry. And what's really, really important, I can't emphasize this enough, is to have the best possible positive and negative controls with which to perform your titration. So that will clearly optimize your signal to noise ratio. And the gold standard is probably a knockout of something. That's not always possible, but there are many ways, and you should really give a lot of thought to the biology and to those positive and negative controls. And this is the PhosphoFlow 101. Again, it's just in there for your reference, but it's the myelomonocytic U937 cell line, some other immune hematopoietic cell lines, the stims, and then the activated pathways. And so you should be able to see this has been designed to give you to really be able to see nice responses here and give you a check on how technically successful you've been. Then, of course, there are the samples, and so cell lines are single cells, especially suspension cells, but there are adherent cells, and so you need to figure out the conditions to get adherence into suspension. There are solid tumors that I'm interested in, so what are the conditions to get viable cells in suspension that you can work with, and we've worked a lot of that out, and there are protocols that I'm very happy to share. And you really want to verify the sample quality. You need viable cells, no good if they're dead. And that can be simply done with trypan blue. And then you can then, again, as part of the background work, you can look at signaling responses. And again, very nice if you can do it with something that's relevant to the biology of the system you're studying. In general, we use 4-volumyristic acetate ionomycin and or pervanidate, and these are non-physiological. I call them pharmacological sledgehammers, because if you don't see anything with them, there's really something wrong. So you really should be able to see profound responses. The pervanidate is a tyrosine phosphatase inhibitor, and so by treating cells with that, all the phosphorylated proteins, the post-translationally modified phosphoproteins, that phospho should persist and remain on the proteins, and so you should have a really nice bright signal. And just to show you an example, not in your handout, about choosing the right clone, this is an example using CD56. And it's looking at it on peripheral blood mononuclear cells. This is fluorescence, and you can see that this clone is able to distinguish bright and dim. And this is actually now on mass cytometry. But what you can see, I mean, clearly the point I want to make is that here are 3 different clones, and these really don't do too much. I'd call them quite dead, but not totally dead. But, and this is the clone you want to go after, and you can see this nice comparable signal between the fluorescence and the mass cytometry. And so that selecting clones, when you've got something new, what I tend to do is I just buy a bunch of clones if I can find them. Sometimes you can ask the vendor if they could just spot you a little bit so you don't have to spend too much money, and sort of beg them, and they often tell them, what it will mean for them and in the future. Just go all dramatic on them. And so now you should be ready for mass cytometry. And so not just to tell you, there are lots and lots of detailed protocols available. You're going to get, have access to them. So, I'm not going to go into it too much. Again, just want to emphasize that a great stopping place if you want to send samples to somebody is the freezing after fixation. So the protocols, again, this is the list slide, and I'll just, pop through it so that you know what to expect, because all my colleagues who will be speaking after me are going to go through these in pretty great detail. So clearly, the surface staining and the intracellular staining, and that should not be any different from fluorescence. Now you get into a mass cytometry specific protocol, namely iridium and rhodium. The intercalator protocol intercalates into the DNA, lets you know you have a cell. Testing pre-conjugated antibodies available from DVS. I don't think we're now Fluoridyme, but I don't think anybody else sells them, right? Nobody else would sell metal conjugated antibodies unless that we do, yeah. And then you want to, of course, after you've done the conjugations yourself, you want to do titrations to check to see how good your antibodies are. IDU treatment for looking at the cell cycle, and then cisplatin acts as a nice viability stain. You've probably heard of cisplatin as a horrid chemotherapeutic agent, but this is a cisplatin pulse, and it reacts very rapidly with proteins. Again, we have a cytometry paper on that, and somebody's going to be talking about that. Again, the clone selection, you can check what clones DVS uses. And then you'll have to, this is a bit repetitive, but you'll have to then do your own conjugations. The panel building, okay, this is really important. These are the factors that go into it. Number one, your protein abundance, so the design of the panel. What metal you put on what protein. The protein abundance, what's the metal enrichment? Is there a metal that particularly has contaminants in it, more contaminants than others, and plus 60 in some of the metals, not very many, have an oxidation potential. And again, I cannot emphasize this enough. Good positive and negative cell controls, you have to get the optimal signal-to-noise ratio. These are, and again, performing the titrations in biological systems that most closely resembles the one you are studying. You can actually do the titrations. You can use multiple antibodies at the same time because there's no overlap fluorescence. And these are examples of some of the cell types that you could use for titrating the antibodies. I put a question mark on primary samples. You probably don't want to do that. What's really nice is to have something then that you use for titrating, and then you can include it in all your subsequent experiments. So you've got you can actually just see how the experiment is working because you keep maintaining the same control. This is an example of a panel. I'm not going to go through all the antibodies, but the points I'm showing you here is this is how we've designed it. Here's the element and its mass. Here are the antibodies, and then what I've done here is was for a collaboration looking at a mouse knockout. And what I've done here is I thought, rather than put all the clone information, is to put in how we titrated everything. And this actually took quite a long time to do. It was a few months. Then I think one thing to appreciate is that you've done all this hard work to get your panel into place. Then you run your first foundational experiment. And I cannot emphasize enough about doing a small foundational experiment. Don't go in and do 50 samples. Just do 6, a few, because then what you will find is that you'll find that some antibodies are not particularly helpful and you wished you'd put in something else. So the first experiments, again, and that takes time, so the first experiments will allow you to sort of tweak the panel. This is really, the panel design is an iterative process, and you hopefully not, when you're too far down the line, think, oh my God, there's something even more interesting. And then I put a little template together because I like to, it's all very personal, but this is how I like to record the antibody titration date. Here's the antigen, here's some of the information. It's obvious, but it's really nice to start collecting that. Then you can have your raw data, and then you can have a little plot, because I'm very visual. I need to see it. Some of the folks in the lab, they can look at the histograms or the contour plots and say, oh, yeah, that looks good, but I actually like to I like to see it. And so here are some real live examples. So here's a titration looking at CD3 in murine splenocytes. And some of the titrations can get quite complex. So clearly you can imagine with cell lines, you just have, you found your good positive and negative control. But when you're using, say, a primary tissue like a splenocyte, you're going to have antibodies that act as counterstains so that you can pull out your T cells and you can look at CD3 or CD8 or CD4 or whatever. And Matt, I'm sure, was going to get into more of that. But this is very, very simple. And what it's just showing you is that you've got a nice titration of CD3 after you gated out. You've been able to gate out these T cells by some other markers, nothing in the B cells. And you see a little blip in the singlets. And the singlets, of course, are the whole population. And that's quite consistent. And then another example looking at an intracellular marker. Here we're looking at phospho-ERK, and we're using PMA, phorbol myristic acetate, which phosphorylates PKC, and that goes right down the RAV-RAS-MET kinase pathway. And so that was one of the positive controls I'd mentioned to you. And as you can see, you've got a nice signal-to-noise ratio. So to sort of tie up, So fluorescence, we like to say it's not over, definitely isn't over. It's incredibly important. It plays a role in these experiments, and it synergizes with CyTOF. And just because you have one or you know someone who has one doesn't mean that you don't do any more hard work. And as I have stressed and emphasized, asking relevant biological questions along with focused foundational work will lead to meaningful data. It's a great, CyTOF is a great screening tool. You can look at your biological landscape and then hone in on things. But really, people ask a lot. I get asked about whether this is going to be in the clinic. And I'd say not yet. It's certainly not an FDA-approved instrument. And then, as I said, after you've looked at your landscape, you can now focus in. The informatics, we have multiple approaches. You're going to hear about some, lots more effort. But the knowledge gain is just amazing when you've really figured out how everything is good, everything's technically sound, and then you get your data. It's a pretty great feeling. I have 6 CyTOFs on campus, but that might be 7, Sean?
6.
Oh, all right.
My lab's the 6th one.
Oh, I thought it was the 7th one. Oh, well, never mind.
2 in Gary's, 2 in the HIMC, 1
Facts.
in the facility.
Oh, you're right. I can't count. Right. I can count up to 40, but I can't count to 6. But and so there they are. This 70 worldwide. I'm not sure. That was my last update about a few months ago, but I do know there's one on every continent. So this is how your time will be divided when you drink the CyTOF Kool-Aid. And I just want you to remember this pie chart when your PIs or whoever, collaborators, come and say, where is the data? And that's the end.
I got thinking when you were talking about your positive and negative controls. So in basic flow cytometry, with and without a stimulation with, say, 2 fluorophores.
Yes.
You end up with 10 tubes.
Yep.
At the positive and negative controls.
Yep.
How does that apply to the mass spec? Does it become 30 times?
No, it doesn't, actually. I think it becomes, thinking of my cell experiments or my negative positive controls with cell lines, I do a 6 to 8 point titration with using the negative and a 6 to 8 point titration with the positive. So that's about 16 tubes, and there's no fluorescence. And it might be that some of the systems, actually you can use the same antibodies, so you actually can do that. You can actually titrate some antibodies. You can depending on the biology, you can titrate them simultaneously.
I can add something to that. You can also, for your positive and negative control, you can also mix your stimulation, and that way you reduce the number of samples.
Yes. Once you know what each stimulation does individually. Yes.
Naively, why wouldn't you validate on the CyTOF just because it's slower than using a fax?
Well, the reason is, because you'd have to use, so if, for example, if you didn't have your, yes, it was definitely one answer. The other is, it's nice to validate, because that's going to be your experiment. It's really nice, my point of view, my personal feeling, to have the actual metal conjugated antibody that you're going to use. And that might not yet be available. So yes, you could go in the CyTOF and you could do maybe a secondary. So there are a number of reasons. Yes, you could, but also, and it is just quicker. If you've got something you've never used before, antibodies you've never used before, or an epitope that's never been measured before, and you get a sort of, five different clones of an antibody, you can very quickly see that on fluorescence, and it'll take longer on CyTOF. So, yeah, a variety of reasons, and there may be some times where you might want to just go straight into the CyTOF. So it's very case dependent.
You could definitely do it on this.
Oh, of course you can.
If it was your only instrument.
Yes. You can absolutely do it, but it's just, practically you get a quick look-see on the fluorescence and you know your answer.
And there's no lineup for the LSR25.
Yes, it's actually a poor little instrument. It's like very lonely.
And as a follow-up, which is maybe better for someone else, is it if you're trying to take a antibody that from flow into Cytop, are you better to buy a pre-conjugated or an unconjugated antibody and test it with a secondary in flow?
It depends, and it doesn't really matter. I've actually done both.
How many antibodies do you think it's possible to titrate simultaneously?
It's a very good question.
Yes.
I don't know
Yeah.
what. I'll tell you the most I've done. I don't even know if I should be admitting this. I think I've titrated like.
Don't admit
I'm
it.
looking at Estrella because we've done some pretty dodgy ones before, like 14 at a time in 1/2, maybe 14, 15. The limit is actually the volume, because even if you're adding like, when you get to your higher concentration, even if you're adding a couple microliters per antibody, it gets to the point where you can't actually fit your cells and all your antibodies in 100 microliters in your tube. Do you know what I mean? You run out of volume, and then, you really, one of the things you'll learn in this course is the staining volume and actual effective antibody concentration is very important.
It's critical.
Right?
It's
You
critical.
This whole, one of the first things I learned when I came to this lab was you actually have to measure the volume of cells you're staining. You can't just suck it down to the line on the tube and then throw some antibody on top of it, because that can cause, when you're doing things like phosphorylation analysis, you're only having 2-3-fold shifts, that can cause huge artifacts in your data. And so when we titrate our antibodies, it's very consistent. Everything's in exactly 100 microliters at a certain cell concentration, same number of cells per tube. But you can certainly multiplex quite a bit in terms of titrating, as long as you have all the right positive and negative control cell populations, like in peripheral blood. You could, use B cells and T cells for positive and negative controls of one another. But then you have to get the counter stains in the tube as well. But certainly, 5 or 6 antibodies at the same time is a big issue.
No more questions. I think is it the break now?
It's the break. Everyone gets coffee for a few minutes.